Good morning, dear students. Welcome to UDAAN Career Institute. In today's class, we will continue with Chapter 2, Class 12 Biology, that is Reproduction in Angiospermic Plants. Now, as you know that in the last class, we have completed microsporogenesis, that is the formation of microspores from pollen mother cells. So, formation of microspores from pollen mother cell was known as microsporogenesis, and this was the diagram which we have gone in the last class, that is the microspore mother cell will undergo meiosis 1 leading to the formation of microspore dyad, and which further undergoes meiosis 2 leading to the formation of a microspore tetrad. All four cells of the microspore tetrad will be haploid in nature. Now, dear students, these are the points which are with respect, which are mainly used for the examination point of view. The largest pollen, it is of the Mirabilis plant. The smallest pollen, it is of Myosotis, and the longest pollen, it is of Zostera, also known as filiform pollen. Now, in today's class, we will go with the structure of pollen grain. Also, it is known as male gametophyte. Now, the study of pollen grain, it is known as palynology. Palynology is also known as study of pollen grains. Now, these pollen grains, they represent the male gametophyte. So, pollen grains are considered as male gametophyte. These are immature male gametophytes. We can also term them as immature male gametophyte. Now, dear students, when you touch the anther or when you open the anther of hibiscus or any other flower, and when you touch the anthers in that, you will see that a yellowish powder, it gets deposited on the tip of your fingers. And when this yellowish powder, when you drop water, or when you sprinkle this yellowish powder on a drop of water, and when you see in a microscope, then you can see that there are variety of sizes, shapes. So in microscope we can see variety of size and shape and color of these pollen grains, and from different species we get different pollen grains, and they all have different size, they all have different shapes as well as colors. So here in the diagram which has been shown below, you can see that there are variety of size, shape and color of the pollen grains. Now, discussing in detail about the structure of pollen grains are spherical in nature with 20 to 50 micrometer in diameter, and it has a two-layered wall. The outer layer is hard, which is known as exine, and the inner layer, it is known as intine. The exine, it is made up of sporopollenin, and this sporopollenin, it is considered as one of the most resistant organic material which is present or which is known till now. Sporopollenin is basically secreted from tapetum. If you remember that there were four layers in case of microsporangia. The innermost layer was the tapetum, and that tapetum, it is secreting sporopollenin. And this sporopollenin, it is one of the most resistant organic material. So exine is made up of sporopollenin. The sporopollenin, it can withstand very high temperatures. Also, it can withstand strong acids and alkali. So it is not getting destroyed by temperature or acids or alkali, and that's why these sporopollenin, they are highly resistant. No enzymes they are present till now who can degrade sporopollenin, and that's why it is because of the sporopollenin that a particular pollen grain, it can be preserved as fossil for a very long duration of time. Now, if you see the exine of pollen grains, we can see that there are prominent apertures in the exine where sporopollenin is absent. These prominent apertures where sporopollenin is absent, it is known as germ pores. So, it is through the germ pores that the pollen tube in future it is coming out during the process of germination. So, germ pores are all those prominent apertures where sporopollenin is absent. Now, exine, it has different patterns as well as designs, and because of this, these pollen grains are having different kind of shapes and structures. The inner wall of the pollen grain is known as intine. It is a thin and continuous layer, and it is made up of cellulose and pectin. So this is an important question for the exam, that what is intine made up of? So intine is made up of cellulose and pectin, and these pollen grains, the cytoplasm is surrounded by a plasma membrane. So once again, I'll repeat the important points. Pollen grains are spherical in nature. They have two layered wall. Outer layer is exine and the inner layer is intine. Exine is made up of sporopollenin. Sporopollenin is one of the most resistant organic material. It cannot be degraded by enzymes. It cannot be degraded by high temperature or acids or alkali. The sporopollenin is secreted by tapetum. Now, exine has prominent apertures where sporopollenin is absent, which is known as germ pores, and due to this, pollen grains are well preserved as fossils. Now, intine is made up of cellulose and pectin, and it is the inner layer of the pollen grains, and the cytoplasm of the pollen grain is surrounded by a plasma membrane. Now, after going through the structure of the pollen grains, next we'll go with the vegetative cell and generative cell. Now, when the pollen grain matures, it contains two cells. That is, one is the vegetative cell and the another one is the germinative cell. So during the maturation, pollen grain contains two types of cells. One is vegetative cell and the another one is generative cell. The vegetative cell is bigger in nature, or it is bigger. It has abundant amount of food reserve material. It is irregular in its shape of the nucleus, while the generative cell, it is smaller and it floats into the cytoplasm of the vegetative cell. And a generative cell, it has a spindle shape and has dense cytoplasm as well as nucleus. So if you compare these two cells, that is vegetative cell and generative cell, the vegetative cell is bigger in nature. It is also known as tube cell, and the another one is known as generative cell. And this generative cell, in future, it is going to give rise to two male gametes. Now, in 60% of the angiosperms, the pollen grains, they are shed from the anthers in two cell stage. That is, it is containing one vegetative cell and it is containing one generative cell. So in two cell stage, when the pollen grain is present in this two cell stage, it gets shed from the anthers. Whereas in the remaining species, the pollen grains are shed in three cell stages. So from anther, it gets shed in three cell stages. Now these three cells are, one will be the vegetative cell, similar to the earlier one, and the second one, the generative cell, it undergoes division, giving rise to two male gametes. As a result, the three cell stage is formed. One is vegetative cell, and there will be formation of two male gametes. Together, it forms a three cell stage. Now, the intine, which is the inner layer of the pollen grains, it will come out through any one of the germ pore. Normally, a pollen grain will be having 3 to 4 germ pores. So, from any one of the germ pore, intine will be coming out in the form of pollen tube during the process of germination of pollen grains. And this pollen tube will be carrying these vegetative cell and two male gametes for the further process of fertilization. Now, the plants in which pollination which is taking place by insect, their pollen grains, they have an oily layer around them. So in those plants where pollination is taking place by insect, their pollen grains have an oily layer around them, which is known as pollen kit. And this pollen kit, it is composed of lipids and carotenoids. It is because of this oily layer or pollen kit that the pollen grains will stick to the insect, and it helps or facilitates in the process of pollination. Now, if you see pollen grains of many species, it causes allergies. These pollen grains are allergic in nature. they can cause bronchial afflictions in some people, and it leads to chronic respiratory disorders like asthma, bronchitis. Now, here is an example of allergic reaction. Now, Parthenium or carrot grass, which came into India along with imported wheat, has become ubiquitous in occurrence. It was responsible for causing pollen allergy. So, Parthenium or carrot grass was causing pollen allergy, and it came into India along with imported wheat. So, this is an example of an allergic reaction of pollen grain. Now, pollen grains are rich in nutrients now, and because of this, nowadays they are mainly used as food supplements. In Western countries, these pollen grains are available in the form of tablets or in the form of syrup, and they are being given to athletes as well as race horses so that their performance can be increased up to a very high extent. So, as a nutritive purpose, it plays a very role in this modern society. Coming to the next part, that is viability of pollen grains. Now, what do you mean by viability? Viability of pollen grain, basically it is a property of pollen grain or ability of pollen grain to undergo fertilization. So basically it is a time span or a period till which the pollen grain has an ability to undergo fertilization. After the viability period, pollen grain will lose its ability for fertilization. So when the pollen grains they are shed from the anthers, so they have to land on the surface of stigma before they lose their viability. So before they lose their viability, these pollen grains should come on the surface of stigma for the process of fertilization. Now, the viability of these pollen grains, it is different in different pollen grain species or different plant species. The period for which the pollen grain remains viable, it is different or highly variable, and it depends on temperature as well as humidity of the atmosphere. In some cereals such as rice and wheat, the viability of pollen grain is 30 minutes. So within 30 minutes, they have to come to the surface of the stigma for the process of fertilization. After 30 minutes, they will lose the viability. Whereas in some members of Rosaceae, Leguminosae and Solanaceae family, their viability is for months. Now, similar to the storing of semen or sperm of many animals, as well as in case of human beings, for artificial insemination, nowadays pollen grains can also be stored in the same way at minus 196 degrees Celsius in liquid nitrogen. So, as we can store semen or sperm of any animal or humans for future use, the same way we can also store these pollen grains in liquid nitrogen at minus 190 degrees Celsius. This is known as cryopreservation. Such pollens can be used as pollen banks, similar to seed banks in case of crop breeding programs. So students, in the next class we will continue with the another topic, that is the female reproductive organ or the gynoecium. Thank you very much.